D. melanogaster had historically been used in laboratories to study genetics and patterns of inheritance. However, D. melanogaster also has importance in environmental mutagenesis research, allowing researchers to study the effects of specific environmental mutagens. 51. Reasons for use in laboratories. D. melanogaster multiple mutants clockwise from top, brown eyes and black cuticle, 2 mutations, cinnabar ice and wild-type cuticle, 1. Mutation, sepia eyes and ebony cuticle, vermilion eyes and yellow cuticle, white eyes and yellow cuticle, wild-type eyes and yellow cuticle. There are many reasons the fruit fly is a popular choice as a model organism. Its care and culture require little equipment, space, and expense even when using large cultures. It can be safely and readily anesthetized, usually with ether, carbon dioxide gas, by cooling, or with products such as FlyNap. Its morphology is easy to identify once anesthetized. It has a short generation time, about 10 days at room temperature, so several generations can be studied within a few weeks. It has a high fecundity, females lay up to 100 eggs per day, and perhaps 2,000 in a lifetime. 12 males and females are readily distinguished, and virgin females can be easily identified by their light-colored, translucent abdomen, facilitating genetic crossing. The mature larva has giant chromosomes in the salivary glands called polyidine chromosomes, puffs, which indicate regions of transcription, hence gene activity. The underreplication of rDNA occurs resulting in only 20% of DNA compared to the brain. Compared to the 47%, less rDNA in Sarcophaga barbata ovaries. It has only 4 pairs of chromosomes, 3 autosomes, and 1 pair of sex chromosomes. Males do not show meiotic recombination, facilitating genetic studies. Recessive lethal, balancer chromosomes, carrying visible genetic markers can be used to keep stocks of lethal alleles in a heterozygous state without recombination due to multiple inversions in the balancer. The development of this organism, from fertilized egg to mature adult, is well understood. Genetic transformation techniques have been available since 1987. One approach of inserting foreign genes into the Drosophila genome involves P elements. The transposable P elements, also known as transposons, are segments of bacterial DNA that are transferred into the fly genome. Transgenic flies have already contributed to many scientific advances, e.g., modeling such human diseases as Parkinson's, neoplasia, obesity, and diabetes. Its complete genome was sequenced and first published in 2000. 52. Sexual mosaics can be readily produced, providing an additional tool for studying the development and behavior of these flies. 53. Genetic markers. D. melanogaster, which carries the psi allele, right, hence showing a characteristic phenotype of curly wings in adult flies. 54. Genetic markers are commonly used in Drosophila research, for example within balancer chromosomes or P-element inserts, and most phenotypes are easily identifiable either with the naked eye or under a microscope. In the list of a few common markers below, the allele symbol is followed by the name of the gene affected and a description of its phenotype. Note: recessive alleles are in lowercase, while dominant alleles are capitalized. Psi 1. Curly, the wings curve away from the body, flight may be somewhat impaired. E1. Ebony, black body and wings. Heterozygotes are also visibly darker than wild type. SV1. Stubble, bristles are shorter and thicker than wild type. W1. White, eyes lack pigmentation and appear white. BW. Brown, eye color determined by various pigments combined. Y1. Yellow, body pigmentation and wings appear yellow, the fly analog of albinism. Classic genetic mutations. Drosophila genes are traditionally named after the phenotype they cause when mutated. For example, the absence of a particular gene in Drosophila will result in a mutant embryo that does not develop a heart. Scientists have thus called this gene Tinman, named after the Oz character of the same name. 55. Likewise, changes in the shaven baby gene cause the loss of dorsal cuticular hairs in Drosophila secelia larva. 56. This system of nomenclature results in a wider range of gene names than in other organisms. B. Black. The black mutation was discovered in 1910 by Thomas Hunt Morgan. 57. The black mutation results in a darker colored body, wings, veins, and segments of the fruit fly's leg. 58. This occurs due to the fly's inability to create beta-alanine, a beta amino acid. 57. The phenotypic expression of this mutation varies based on the genotype of the individual. For example, whether the specimen is homozygotic or heterozygotic results in a darker or less dark appearance. 58. This genetic mutation is X-linked recessive. BW. Brown eye mutation results from inability to produce or synthesize pteridine, red pigments, due to a point mutation on chromosome 2. 60. M. Miniature. One of the first records of the miniature mutation of wings was also made by Thomas Hunt Morgan in 1911. He described the wings as having a similar shape as the wild-type phenotype. However, their miniature designation refers to the lengths of their wings, which do not stretch beyond their body and, thus, are notably shorter than the wild-type length. He also noted its inheritance is connected to the sex of the fly and could be paired with the inheritance of other sex-determined traits such as white eyes. 61. The wings may also demonstrate other characteristics deviant from the wild-type wing, such as a duller and cloudier color. 62. Miniature wings are 1.5x shorter than wild-type but are believed to have the same number of cells. This is due to the lack of complete flattening by these cells, making the overall structure of the wing seem shorter in comparison. The pathway of wing expansion is regulated by a signal receptor pathway, where the neurohormone bursicon interacts with its complementary G protein coupled receptor. This receptor drives one of the G protein subunits to signal further enzyme activity and results in development in the wing, such as apoptosis and growth. 63. Say, sepia. The eye color of the sepia mutant is sepia, a reddish-brown color. In wild-type flies, omochromes brown and drossopterans red give the eyes the typical red color. 64-65 The drossopterans are made via a pathway that involves a pyrimidodiazepine synthase, 66, which is encoded on chromosome 3L. The gene has a premature stop codon in sepia flies, so that the flies cannot produce the pyrimidodiazepine synthase and thus no red pigment, so that the eyes stay sepia. 64 The sepia allele is recessive and thus offspring from sepia flies and homozygous wild-type flies has red eyes. The sepia phenotype does not depend on the sex of the fly. 67b Vermilion mutants cannot produce the brown omochromes, leaving the red drossopterans so that the eyes are vermilion colored, a radiant red compared to a wild-type D. melanogaster. The vermilion mutation is sex-linked and recessive. The gene that is defect lies on the X chromosome. 68 The brown omochromes are synthesized from kynurenine, which is made from tryptophan. Vermilion flies cannot convert tryptophan into kynurenine and thus cannot make omochromes, either. 68 Vermilion mutants live longer than wild-type flies. This longer lifespan may be associated with the reduced amount of tryptophan converted to kynurenine in vermilion flies. 69. Triple mutant male fruit fly. Drosophila melanogaster, exhibiting black body, vestigial wings, and brown eyes mutations. VG, vestigialis spontaneous mutation, discovered in 1919 by Thomas Morgan and Calvin Bridges. Vestigial wings are those not fully developed and that have lost function. Since the discovery of the vestigial gene in Drosophila melanogaster, there have been many discoveries of the vestigial gene in other vertebrates and their functions within the vertebrates. 70 The vestigial gene is considered to be one of the most important genes for wing formation, but when it becomes overexpressed, the issue of ectopic wings begin to form. 71 The vestigial gene acts to regulate the expression of the wing imaginal discs in the embryo and acts with other genes to regulate the development of the wings. A mutated vestigial allele removes an essential sequence of the DNA required for correct development of the wings. 72 W. White Drosophila melanogaster wild type typically expresses a brick red eye color. The white eye mutation in fruit flies is caused due to the absence of two pigments associated with red and brown eye colors, peridines red and omochromes brown. 65 In January 1910, Thomas Hunt Morgan first discovered the white gene and denoted it as W. The discovery of the white eye mutation by Morgan brought about the beginnings of genetic experimentation and analysis of Drosophila melanogaster. Hunt eventually discovered that the gene followed a similar pattern of inheritance related to the meiotic segregation of the X chromosome. He discovered that the gene was located on the X chromosome with this information. This led to the discovery of sex-linked genes and also to the discovery of other mutations in Drosophila melanogaster. 73 The white eye mutation leads to several disadvantages in flies, such as a reduced climbing ability, shortened lifespan, and lowered resistance to stress when compared to wild-type flies. 74 Drosophila melanogaster has a series of mating behaviors that enable them to copulate within a given environment and therefore contribute to their fitness. After Morgan's discovery of the white eye mutation being sex-linked, a study led by Sturtevant, 1915, concluded that white-eyed males were less successful than wild-type males in terms of mating with females. 75 It was found that the greater the density in eye pigmentation, the greater the success in mating for the males of Drosophila melanogaster. 75 Y. Yellow gene is a genetic mutation known as DMEL-Y within the widely used database called flybase. This mutation can be easily identified by the atypical yellow pigment observed in the cuticle of the adult flies and the mouthpieces of the larva. 76 The Y mutation comprises the following phenotypic classes: the mutants that show a complete loss of pigmentation from the cuticle Y type, and other mutants that show a mosaic pigment pattern with some regions of the cuticle wild type, Y2 type. 77 The role of the yellow gene is diverse and is responsible for changes in behavior, sex-specific reproductive maturation and epigenetic reprogramming. 78 The Y gene is an ideal gene to study as it is visibly clear when an organism has this gene, making it easier to understand the passage of DNA to offspring. 78 Wild-type wing left versus miniature wing right. Genome. The genome of D. melanogaster, sequenced in 2000, and curated at the flybase database, 52 contains 4 pairs of chromosomes, an XY pair, and 3 autosomes labeled 2, 3, and 4. The 4th chromosome is relatively very small and therefore often ignored, aside from its important eyeless gene. The D. melanogaster sequence genome of 139.5 million base pairs has been annotated, 79, and contains around 15,682 genes according to Ensemble Release 73. More than 60% of the genome appears to be functional nonprotein coding DNA, 80 involved in gene expression control. Determination of sex in Drosophila occurs by the X-A ratio of X chromosomes to autosomes, not because of the presence of a Y chromosome as in human sex determination. Although the Y chromosome is entirely heterochromatic, it contains at least 16 genes, many of which are thought to have malarial-related functions. 81. There are 3 transferrin orthologs, all of which are dramatically divergent from those known in short-8 models. 82. Similarity to humans. A June 2001 study by National Human Genome Research Institute comparing the fruit fly and human genome estimated that about 60% of genes are conserved between the 2 species. 83. About 75% of known human disease genes have A recognizable match in the genome of fruit flies. 84 and 50% of fly protein sequences have mammalian homologs. An online database called Homophila is available to search for human disease gene homologs in flies and vice versa. 85. Drosophila is being used as a genetic model for several human diseases including the neurodegenerative disorders Parkinson's, Huntington's, spinocerebellar ataxia and Alzheimer's disease. 86. The fly is also being used to study mechanisms underlying aging and oxidative stress, immunity, diabetes, and cancer, as well as drug abuse. 87, 88, 89. The life cycle of this insect has 4 stages: fertilized egg, larva, pupa, and adult. 14. Genomic information. D. Melanogaster chromosomes to scale with megabase pair references oriented is in the National Center for Biotechnology Information Database. IEW/map_search.cgi. Taxit equals 7,227. Centimorgan distances are approximate and estimated from the locations of selected mapped loci. NCBI genome ID 47. nlm.ni.gov/genome. Term equals 4. Embryo development. Development. Embryogenesis in Drosophila has been extensively studied, as its small size, short generation time, and large brood size makes it ideal for genetic studies. It is also unique among model organisms in that cleavage occurs in a syncytium. D. Melanogaster oogenesis. During oogenesis, cytoplasmic bridges called ring canals connect the forming oocyte to nurse cells. Nutrients and developmental control molecules move from the nurse cells into the oocyte. In the figure to the left, the forming oocyte can be seen to be covered by follicular support cells. After fertilization of the oocyte, the early embryo, or syncytial embryo, undergoes rapid DNA replication and 13 nuclear divisions until about 5,000 to 6,000 nuclei accumulate in the unseparated cytoplasm of the embryo. By the end of the 8th division, most nuclei have migrated to the surface, surrounding the yolk sac, leaving behind only a few nuclei, which will become the yolk nuclei. After the 10th division, the pole cells form at the posterior end of the embryo, segregating the germ line from the syncytium. Finally, after the 13th division, cell membranes slowly invaginate, dividing the syncytium into individual somatic cells. Once this process is completed, gastrulation starts. 90. Nuclear division in the early Drosophila embryo happens so quickly, no proper checkpoints exist, so mistakes may be made in division of the DNA. To get around this problem, the nuclei that have made a mistake detach from their centrosomes and fall into the center of the embryo yolk sac, which will not form part of the fly. The gene network transcriptional and protein interactions governing the early development of the fruit fly embryo is one of the best understood gene networks to date, especially the patterning along the anteroposterior AP and dorsoventral DV axes, see under morphogenesis. 90. The embryo undergoes well-characterized morphogenic movements during gastrulation and early development, including germ band extension, formation of several furrows, ventral invagination of the mesoderm, and posterior and anterior invagination of endoderm gut, as well as extensive body segmentation until finally hatching from the surrounding cuticle into a first instar larva. During larval development, tissues known as imaginal discs grow inside the larva. Imaginal discs develop to form most structures of the adult body, such as the head, legs, wings, thorax, and genitalia. Cells of the imaginal discs are set aside during embryogenesis and continue to grow and divide during the larval stages, unlike most other cells of the larva, which have differentiated to perform specialized functions and grow without further cell division. At metamorphosis, the larva forms a pupa, inside which the larval tissues are reabsorbed and the imaginal tissues undergo extensive morphogenic movements to form adult structures. Developmental plasticity. Biotic and abiotic factors experienced during development will affect developmental resource allocation, leading to phenotypic variation, also referred to as developmental plasticity. 91, 92, as in all insects, 92, environmental factors can influence several aspects of development in Drosophila melanogaster. 93, 94, fruit flies reared under a hypoxia treatment experience decreased thorax length, while hyperoxia produces smaller flight muscles, suggesting negative developmental effects of extreme oxygen levels. 95, circadian rhythms are also subject to developmental plasticity. Light conditions during development affect daily activity patterns in Drosophila melanogaster, where flies raised under constant dark or light are less active as adults than those raised under a 12-hour light-dark cycle. 96. Temperature is one of the most pervasive factors influencing arthropod development. In Drosophila melanogaster, temperature-induced developmental plasticity can be beneficial and/or detrimental. 97, 98, most often lower developmental temperatures reduce growth rates, which influence many other physiological factors. 99, for example, development at 25 degrees Celsius increases walking speed, thermal performance, breadth, malperformance curve relationship between environmental _fig 1 221,716,210, and territorial success, while development at 18 degrees Celsius increases body mass, wing size, all of which are tied to fitness. 94, 97, moreover, developing at certain low temperatures produces proportionally large wings which improve flight and reproductive performance at similarly low temperatures, see acclimation. 100. While certain effects of developmental temperature, like body size, are irreversible in ectotherms, others can be reversible. 92. 101. When Drosophila melanogaster develop at cold temperatures, they will have greater cold tolerance, but if cold-reared flies are maintained at warmer temperatures, their cold tolerance decreases and heat tolerance increases over time. 101. 102. Because insects typically only mate in a specific range of temperatures, their cold heat tolerance is an important trait in maximizing reproductive output. 103. While the traits described above are expected to manifest similarly across sexes, developmental temperature can also produce sex-specific effects in D. melanogaster adults. Females ovariole number is significantly affected by developmental temperature in D. melanogaster. 104. Egg size is also affected by developmental temperature, and exacerbated when both parents develop at warm temperatures, see maternal effect. 97. Under stressful temperatures, these structures will develop to smaller ultimate sizes and decrease a female's reproductive output. 104. 97. Early fecundity, total eggs laid in first 10 days post-eclosion, is maximized when reared at 25 degrees Celsius versus 17 degrees Celsius and 29 degrees Celsius, regardless of adult temperature. 105. Across a wide range of developmental temperatures, females tend to have greater heat tolerance than males. 106. Malstressful developmental temperatures will cause sterility in D. melanogaster males, although the upper temperature limit can be increased by maintaining strains at high temperatures, see acclimation. 98. Male sterility can be reversible if adults are returned to an optimal temperature after developing at stressful temperatures. 107. Male flies are smaller and more successful at defending food, oviposition sites when reared at 25 degrees Celsius versus 18 degrees Celsius, thus smaller males will have increased mating success and reproductive output. 94. Sex determination. Drosophila flies have both X and Y chromosomes, as well as autosomes. Unlike humans, the Y chromosome does not confer maleness, rather, it encodes genes necessary for making sperm. Sex is instead determined by the ratio of X chromosomes to autosomes. 108. Furthermore, each cell decides whether to be male or female independently of the rest of the organism, resulting in the occasional occurrence of gynandromorphs. X chromosomes autosomes ratio of X, A. 3 major genes are involved in determination of Drosophila sex. These are sex-lethal, sisterless, and deadpan. Deadpan is an autosomal gene which inhibits sex-lethal, while sisterless is carried on the X chromosome and inhibits the action of deadpan. An AAX cell has twice as much deadpan as sisterless, so sex-lethal will be inhibited, creating a male. However, an AAXX cell will produce enough sisterless to inhibit the action of deadpan, allowing the sex-lethal gene to be transcribed to create a female. Later, control by deadpan and sisterless disappears, and what becomes important is the form of the sex-lethal gene. A secondary promoter causes transcription in both males and females. Analysis of the cDNA has shown that different forms are expressed in males and females. Sex-lethal has been shown to affect the splicing of its own mRNA. In males, the third exon is included which encodes a stop codon, causing a truncated form to be produced. In the female version, the presence of sex-lethal causes this exon to be missed out. The other 7 amino acids are produced as a full peptide chain, again giving a difference between males and females. 109. Presence or absence of functional sex-lethal proteins now go on to affect the transcription of another protein known as double sex. In the absence of sex-lethal, double sex will have the fourth exon removed and be translated up to and including exon 6 DSXM, ale, while in its presence the fourth exon which encodes a stop codon will produce a truncated version of the protein, DSXF, a male. DSXF causes transcription of yolk proteins 1 and 2 in somatic cells, which will be pumped into the oocyte on its production. Immunity. The D. melanogaster immune system can be divided into 2 responses, humoral and cell-mediated. The former is a systemic response mediated in large part through the TOL and IMD pathways, which are parallel systems for detecting microbes. Other pathways including the stress response pathways JAK-STAT and P38, nutritional signaling via FOXO, and JNK cell death signaling are all involved in key physiological responses to infection. D. melanogaster has an organ called the fat body, which is analogous to the human liver. The fat body is the primary secretory organ and produces key immune molecules upon infection, such as serine proteases and antimicrobial peptides, AMPs. AMPs are secreted into the hemolymph and bind infectious bacteria and fungi, killing them by forming pores in their cell walls or inhibiting intracellular processes. The cellular immune response instead refers to the direct activity of blood cells, hemocytes, in Drosophila, which are analogous to mammalian monocytes, macrophages. Hemocytes also possess a significant role in mediating humoral immune responses such as the melanization reaction. 110. The immune response to infection can involve up to 2,423 genes, or 13.7% of the genome. Although the fly's transcriptional response to microbial challenge is highly specific to individual pathogens, Drosophila differentially expresses a core group of 252 genes upon infection with most bacteria. This core group of genes is associated with gene ontology categories such as antimicrobial response, stress response, secretion, neuron-like, reproduction, and metabolism among others. 111. 112. Drosophila also possesses several immune mechanisms to both shape the microbiota and prevent excessive immune responses upon detection of microbial stimuli. For instance, secreted PGRPs with amidase activity scavenge and degrade immunostimulatory DAP-type PGN in order to block IMD activation. 113. Unlike mammals, Drosophila have innate immunity but lack an adaptive immune response. However, the core elements of this innate immune response are conserved between humans and fruit flies. As a result, the fruit fly offers a useful model of innate immunity for disentangling genetic interactions of signaling and effector function, as flies do not have to contend with interference of adaptive immune mechanisms that could confuse results. Various genetic tools, protocols, and assays make Drosophila a classical model for studying the innate immune system. 114, which has even included immune research on the International Space Station. 115. JAK-STAT signaling. Multiple elements of the Drosophila JAK-STAT signaling pathway bear direct homology to human JAK-STAT pathway genes. JAK-STAT signaling is induced upon various organismal stresses such as heat stress, dehydration, or infection. JAK-STAT induction leads to the production of a number of stress response proteins including thioester-containing proteins, TEPs, 116, pterandates, 117, and the putative antimicrobial peptide listericin. 118. The mechanisms through which many of these proteins act is still under investigation. For instance, the TEPs appear to promote phagocytosis of gram-positive bacteria and the induction of the toll pathway. As a consequence, flies lacking TEPs are susceptible to infection by toll pathway challenges. 116. Drosophila hemocytes, green, engulfing Escherichia coli bacteria, red. The cellular response to infection. Circulating hemocytes are key regulators of infection. This has been demonstrated both through genetic tools to generate flies lacking hemocytes, or through injecting microglass beads or lipid droplets that saturate hemocyte ability to phagocytose a secondary infection. 119. 120. Flies treated like this fail to phagocytose bacteria upon infection, and are correspondingly susceptible to infection. 121. These hemocytes derive from two waves of hematopoiesis, one occurring in the early embryo and one occurring during development from larva to adult. 122. However, Drosophila hemocytes do not renew over the adult lifespan, and so the fly has a finite number of hemocytes that decrease over the course of its lifespan. 123. Hemocytes are also involved in regulating cell cycle events and apoptosis of aberrant tissue, e.g. cancerous cells, by producing IGR, a tumor necrosis factor signaling molecule that promotes JNK signaling and ultimately cell death and apoptosis. 124. Behavioral genetics and neuroscience. In 1971, Ron Konopka and Seymour Benzer published Clock Mutants of Drosophila melanogaster, a paper describing the first mutations that affected an animal's behavior. Wild-type flies show an activity rhythm with a frequency of about a day, 24 hours. They found mutants with faster and slower rhythms, as well as broken rhythms, flies that move and rest in random spurts. Work over the following 30 years has shown that these mutations, and others like them, affect a group of genes and their products that form a biochemical or biological clock. This clock is found in a wide range of fly cells, but the clock-bearing cells that control activity are several dozen neurons in the fly's central brain. Since then, Benzer and others have used behavioral screens to isolate genes involved in vision, olfaction, audition, learning, memory, courtship, pain, and other processes, such as longevity. Following the pioneering work of Alfred Henry Sturtevant, 125, and others, Benzer and colleagues, 53, used sexual mosaics to develop a novel fate mapping technique. This technique made it possible to assign a particular characteristic to a specific anatomical location. For example, this technique showed that male courtship behavior is controlled by the brain. 53. Mosaic fate mapping also provided the first indication of the existence of pheromones in this species. 126. Males distinguish between conspecific males and females in direct persistent courtship preferentially toward females thanks to a female-specific sex pheromone which is mostly produced by the female stergites. The first learning and memory mutants dunce, rutabaga, etc. were isolated by William Chip Quinn while in Benzer's lab, and were eventually shown to encode components of an intracellular signaling pathway involving cyclic AMP, protein kinase A, and a transcription factor known as CREB. These molecules were shown to be also involved in synaptic plasticity in aplysia and mammals. 127. The Nobel Prize in Physiology or Medicine for 2017 was awarded to Jeffrey C. Hall, Michael Rosbash, Michael W. Young for their works using fruit flies in understanding the molecular mechanisms controlling the circadian rhythm. 128. Male flies sing to the females during courtship using their wings to generate sound, and some of the genetics of sexual behavior have been characterized. In particular, the fruitless gene has several different splice forms, and male flies expressing female splice forms have female-like behavior and vice versa. The TRP channels NAMPC, Nanchung, and inactive are expressed in sound-sensitive Johnston's organ neurons and participate in the transduction of sound. 129. 130. Mutating the genderblend gene, also known as CG6070, alters the sexual behavior of Drosophila, turning the flies bisexual. 131. Flies use a modified version of Bloom filters to detect novelty of odors, with additional features including similarity of novel odor to that of previously experienced examples, and time elapsed since previous experience of the same odor. 132. Aggression. As with most insects, aggressive behaviors between male flies commonly occur in the presence of courting a female and when competing for resources. Such behaviors often involve raising wings and legs towards the opponent and attacking with the whole body. 133. Thus, it often causes wing damage, which reduces their fitness by removing their ability to fly and mate. 134. Acoustic communication. In order for aggression to occur, male flies produce sounds to communicate their intent. A 2017 study found that songs promoting aggression contain pulses occurring at longer intervals. 135 RNA sequencing from fly mutants displaying over-aggressive behaviors found more than 50 auditory-related genes, important for transient receptor potentials, CA2+ signaling, and mechanoreceptor potentials, to be upregulated in the AB neurons located in Johnston's organ. 135. In addition, aggression levels were reduced when these genes were knocked out via RNA interference. 135. This signifies the major role of hearing as a sensory modality in communicating aggression. Pheromone signaling. Other than hearing, another sensory modality that regulates aggression is pheromone signaling, which operates through either the olfactory system or the gustatory system depending on the pheromone. 136. An example is CVA, an anti-aphrodisiac pheromone used by males to mark females after copulation and to deter other males from mating. 137. This male-specific pheromone causes an increase in male-male aggression when detected by another male's gustatory system. 136. However, upon inserting a mutation that makes the flies irresponsive to CVA, no aggressive behaviors were seen. 138. This shows how there are multiple modalities for promoting aggression in flies. Competition for food. Specifically, when competing for food, aggression occurs based on amount of food available and is independent of any social interactions between males. 139. Specifically, sucrose was found to stimulate gustatory receptor neurons, which was necessary to stimulate aggression. 139. However, once the amount of food becomes greater than a certain amount, the competition between males lowers. 139. This is possibly due to an overabundance of food resources. On a larger scale, food was found to determine the boundaries of a territory since flies were observed to be more aggressive at the food's physical perimeter. Effect of sleep deprivation. However, like most behaviors requiring arousal and wakefulness, aggression was found to be impaired via sleep deprivation. Specifically, this occurs through the impairment of octopamine and dopamine signaling, which are important pathways for regulating arousal in insects. 140. 141. Due to reduced aggression, sleep-deprived male flies were found to be disadvantaged at mating compared to normal flies. 141. However, when octopamine agonists were administered upon these sleep-deprived flies, aggression levels were seen to be increased and sexual fitness was subsequently restored. 141. Therefore, this finding implicates the importance of sleep in aggression between male flies. Vision. Stereo images of the eye. The compound eye of the fruit fly contains 760 unit eyes or ommatidia, and are one of the most advanced among insects. Each ommatidium contains 8 photoreceptor cells, R18, support cells, pigment cells, and a cornea. Wild-type flies have reddish pigment cells, which serve to absorb excess blue light so the fly is not blinded by ambient light. Eye color genes regulate cellular vesicular transport. The enzymes needed for pigment synthesis are then transported to the cell's pigment granule, which holds pigment precursor molecules. 65. View from above. Frontal view. Each photoreceptor cell consists of 2 main sections, the cell body and the rhabdomere. The cell body contains the nucleus, while the 100 micrometer long rhabdomere is made up of toothbrush-like stacks of membrane called microvilli. Each microvillus is 1 to 2 μm in length and about 60 nanometers in diameter. 142. The membrane of the rhabdomere is packed with about 100 million opsin molecules, the visual protein that absorbs light. The other visual proteins are also tightly packed into the microvilli, leaving little room for cytoplasm. Opsins and spectral sensitivity. The arrangement of the photoreceptor cells in an pale and yellow ommatidia of Drosophila melanogaster. The top row shows 2 of the 6 outer photoreceptor cells, R1, R6, and the inner R7 and R8 cells. The bottom row shows the different opsins, RH1, RH3, RH4, RH5, and RH6 the cells express. Figure from Sharkey et al. 2020. 143. Expression of the opsin RH1 in the photoreceptor cells R1, R6. The genome of Drosophila encodes 7 opsins, 144, 5 of those are expressed in the ommatidia of the eye. The photoreceptor cells R1, R6 express the opsin RH1, 145, which absorbs maximally blue light, around 480 nanometers, 146, 147, 148. However, the R1, R6 cells cover a broader range of the spectrum than an opsin would allow due to a sensitizing pigment, 149, 150, that adds 2 sensitivity maxima in the UV range, 355 and 370 nanometers. 148. The R7 cells come in 2 types with yellow and pale rhabdomers, R7Y and R7P. 151, 152. The pale R7P cells express the opsin RH3, 153, 154, which maximally absorbs UV light, 345 nanometers. 155. The R7P cells are strictly paired with the R8P cells that express RH5, 154, which maximally absorbs violet light, 437 nanometers. 148. The yellow R7Y cells express a blue-absorbing screening pigment, 151, and the opsin RH4, 156, which maximally absorbs UV light, 375 nanometers. 155. The R7Y cells are strictly paired with R8Y cells that express RH6, 157, which maximally absorbs UV light, 508 nanometers. (171) Grooming. Drosophila are known to exhibit grooming behaviors that are executed in a predictable manner. Drosophila consistently begin a grooming sequence by using their front legs to clean the eyes, then the head and antennae. Using their hind legs, Drosophila proceed to groom their abdomen, and finally the wings and thorax. Throughout this sequence, Drosophila periodically rub their legs together to get rid of excess dust and debris that accumulates during the grooming process. 172. Grooming behaviors have been shown to be executed in a suppression hierarchy. This means that grooming behaviors that occur at the beginning of the sequence prevent those that come later in the sequence from occurring simultaneously, as the grooming sequence consists of mutually exclusive behaviors. 173. 174. This hierarchy does not prevent Drosophila from returning to grooming behaviors that have already been accessed in the grooming sequence. 173. The order of grooming behaviors in the suppression hierarchy is thought to be related to the priority of cleaning a specific body part. For example, the eyes and antennae are likely executed early on in the grooming sequence to prevent debris from interfering with the function of D. melanogaster's sensory organs. 173. 174. Walking. Top view of a walking Drosophila, left with legs tracked with deep cut. 175. Right. Like many other hexapod insects, Drosophila typically walk using a tripod gait. 176. This means that 3 of the legs swing together while the other 3 remain stationary, or in stance. Specifically, the middle leg moves in phase with the contralateral front and hind legs. However, variability around the tripod configuration exists along a continuum, meaning that flies do not exhibit distinct transitions between different gaits. 177. At fast walking speeds, the walking configuration is mostly tripod, 3 legs in stance, but at slower walking speeds, flies are more likely to have 4 tetrapod, or 5 legs in stance wave. 178. 179. These transitions may help to optimize static stability. 180. Because flies are so small, inertial forces are negligible compared with the elastic forces of their muscles and joints or the viscous forces of the surrounding air. 181. Flight. Flies fly via straight sequences of movement interspersed by rapid turns called saccades. 182. During these turns, a fly is able to rotate 90 degrees in less than 50 milliseconds. 182. Characteristics of Drosophila flight may be dominated by the viscosity of the air, rather than the inertia of the fly body, but the opposite case with inertia as the dominant force may occur. 182. However, subsequent work showed that while the viscous effects on the insect body during flight may be negligible, the aerodynamic forces on the wings themselves actually cause fruit flies' turns to be damped viscously. 183. Connectome. Drosophila is one of the few animals, C. elegans being another, where detailed neural circuits, a connectome, are available. A high-level connectome, at the level of brain compartments and interconnecting tracts of neurons, exists for the full fly brain. 184. A version of this is available online. 185. Detailed circuit-level connectomes exist for the lamina, 186, 187, and a medulla, 188 column, both in the visual system of the fruit fly, and the alpha lobe of the mushroom body. 189. In May 2017, a paper published in Bioarxiv presented an electron microscopy image stack of the whole adult female brain at synaptic resolution. The volume is available for sparse tracing of selected circuits. 190. 191. Since then, multiple datasets have been collected including a dense connectome of half the central brain of Drosophila in 2020, 192, 193, and a dense connectome of the entire female adult nerve cord in 2021. 194. Generally, these datasets are acquired by sectioning the tissue, e.g. the brain, into thin sections, on order of 10 or hundreds of nanometers. Each section is then imaged using an electron microscope and these images are stitched and aligned together to create a 3D image volume. The methods used in reconstruction and initial analysis of the such datasets followed. 195. Due to advancements in deep learning, automated methods for image segmentation have made large-scale reconstruction providing dense reconstructions of all the neurites within the volume.